Hi guys, welcome to today's week 7 lecture. Not a lot of slides this week, but there's some juicy slides in here, so I think this should be plenty for everybody. So where have we been? Last time we talked about Tennessee and Kentucky, and we said specifically those were, I don't know what you call them, kind of acronyms to remember the S3 and the S4 heart sound. Remember the S3 heart sound, that was a ventricular gallop, and it sounded like long, short, short, long, short, short, lup, dup, dup, lup, dup, dup, like a galloping horse. Somebody said it sounds like Kentucky, Kentucky. Very worrisome in patients over the years of 40, or over the age of 40 years. Typically indicates aortic stenosis. The S4 heart sound is the atrial gallop. It's just the opposite. It's a short, short, long, short, short, long. And that is bad at all age groups, and it typically indicates left ventricular hypertrophy on the road to heart failure. Both of these are auscultated in the left lateral decubitus position at the mitral auscultation area. So we talked about that in detail. We also got into this dreaded thing, the Wigger's diagram. We didn't go through all of it, but we looked at the ventricular volume curve of the Wigger's, and this is actually 2 beats of the heart it's showing here. And we said that if you follow this pink volume curve, and this is the volume of blood in the left ventricle, we can see that right here at the start of S1, the ventricle is filled up completely, 130 milliliters of blood. And when the heart contracts, it squeezes blood out of the heart, and so you see the volume in the ventricle dropping because the blood is flying up the ascending aorta, or if you're the right ventricle, the pulmonary trunk. This is geared on the left ventricle, though. And we said down here at the S2 heart sound, basically all the blood is out of the heart, and that corresponds with the start of diastole, and the S1 corresponds with the start of systole. And we said that as the ventricle fills up with blood, it does so in 3 phases, basically, 3 1/3. The first third is the rapid filling phase, and if you're going to hear an S3 heart sound, this is where it's going to occur. And then we have the second third, and the third third, that's where ventricular systole occurs. And if you're going to have a pathological S4 sound, that's where it's going to be. It's going to be in this last 1/3 of the filling phase of the cardiac cycle. All right, so make sure you know that. We've already talked about that in depth. We have not talked about the natriuretic peptides at all. And we have talked about the renin-angiotensin-aldosterone system, and this is the opposite of it. The R2A system will restore hypotension to normal, or it'll try to do so. This one's just the opposite. If the blood pressure goes too high, this is what brings it back down to earth, back to that equilibrium. So it's kind of like a teeter-totter with the R2A system on one side, natriuretic system on the other side. That's how you pronounce it, like na, j, tree, u, ret, like bet, ret, ick, natriuretic system. And it's made up of natriuretic peptides, also known as natriuretic factors. And they flap these 2 words back and forth all the time. There's 3 of them that we need to talk about, but again, its main job is to combat hypertension, or they might say to combat hypervolemia, which is going to cause hypertension. That's too much volume in the blood, which causes the hypertension. So that's what it does. Again, its actions are the opposite of angiotensin II. So its job is to decrease blood pressure and decrease blood volume. There's 3 of them, and there's 3 of these natriuretic peptides. And 2 of them are released from the heart. That's an error there. These are brand new slides right here, so make sure you fix that. So not all of them are released. The first 2 are released from the heart. The second is not, and we'll look at that in a little while, just a little bit. They don't know that much about that third one. The big one is the first one. So bottom line, yeah, the heart is an endocrine organ. All right, let's meet the three. So we have the big one that we know most about, and the first one discovered was ANP or ANF, atrial natriuretic peptide or factor. Then we have brain natriuretic peptide or factor. And then we don't know much about this one. This is C type or type C natriuretic peptide or CNP. They're all made by the same gene, and just proteases cleave them at different spots in the same gene. The gene is actually on 2 different chromosomes, though, as we'll see later. All right, let's talk about atrial peptide, ANP. Again, atrial factor. And there's all the AKAs again. We already said that, ANF, ANP. I tend to call it ANP. It was discovered back in 1984 while I was in chiropractic college, or actually just starting chiropractic college way back in the Stone Age. Yep, by far the most studied of the 3. And this one is a true circulating hormone. There's no question about it. It has hormonal activity, and it's made in the right and the left myocardial sites of the atria. So it's made in the muscular part of the atria. More so, just a smidge more. They think more in the right than the left, but some authors say they're the same. But it's definitely both atria that can produce them. How is it made? So an atriomyocardial site, stretch sets it off. We won't get too into the weeds on that mechanism, but it sets off a secondary messenger system that tells the gene for, let's just use ANP here, to go ahead and run through transcription, translation, and make ourselves the ANP. But it's not an active hormone at first. In fact, it's in something called, just like insulin, exactly like insulin, the pre-pro-ANP stage. And that is kicked into the rough endoplasmic reticulum, as all proteins do, almost all of them, after they're made. And the leader gets chopped off, and that creates, we won't worry about the mechanism there, but we are in the rough endoplasmic reticulum, and the front part of it, some call the leader the signaling part, gets chopped off, and it leaves us a pro-ANP product, which is still inactive. And next, the pro-ANP moves to the Golgi apparatus, where it is acted on by a big enzyme called chorin. Definitely need to know chorin. Where is it found? In the Golgi apparatus of atrial myocardial sites. And what does it do? It cuts it in a certain way, does a little chopping here, and it leaves us with this little piece right here. And that is the mature natriuretic peptide or natriuretic factor. And yep, that's how it's created. Very, very similar to insulin. When we get into endocrinology, we'll talk about the same story. So you might as well know it now. And the AMP is released by the Golgi into these little secretory vesicles, and they bind to the plasma membrane, and they get released into the interstitium and sucked into the distal capillary. That usual story. So what stimulates its release? Again, it's the stretch of the cardiac atriomyocyte. What is the stretch from? Anything that increases blood volume in the atria. It's usually from hypertension or hypervolemia. There's other stimuli that can cause this, but usually it's from hypertension. Okay, so yeah, the heart is our main pump in our body that keeps us alive, but it also acts as an endocrine organ, or an endocrine gland, you could even say. Okay, secretes 95% of all the ANP, so a little bit comes from the ventricles, and in the brain they think they might have found a little bit, but by far the lion's share comes from the ventricle, or for the atria. That's why it got the name atrial natriuretic peptide. So what does it do? Well, we already said its job is to lower hypertension. So it does so in a couple of ways, and I'll try to follow my slides along here. We've looked at this before, right, when we talked about the R2A system. So it really does just the opposite of what angiotensin II does, Remember, angiotensin 2 is the main effector of the R2A system. So it has the opposite effect. So here it is down here. There's ANP. So one thing it can do, if it wants to do the opposite of angiotensin 2, it can block the V2 receptor. So vasopressin can't bind. That's one theory anyway. Another one, it goes into, because it has a receptor called the NPRA receptor, atrial natriuretic peptide, or the natriuretic peptide A receptor is also on principal cell membrane. It can bind to that, and there's a secondary messenger system. And we think that can also mainly mess up this pathway right here, kind of decrease the expression of the gene. So the story's not fully understood, but this is what I'm going to test you on right here. So ANP will bind to, and this has been demonstrated in laboratory and experiments before. So ANP binds to the V2 receptor and blocks the function of vasopressin. Remember what vasopressin does. So that's the water, bring in the water hormone, also known as antidiuretic hormone or arginine vasopressin. So if you block that, you're not going to get aquaporin's 2 channels implanted into this apical membrane of the principal cell. And therefore, there's no way for water to get out if these aquaporin's 2 channels aren't there. So that will definitely stop or start decreasing the hypertension, because water is going to be ejected in the glomerulus. And normally water is ejected there and it travels through the renal tubules. And normally we have some water coming back because there's always a little bit of vasopressin around. So these aquaporin's 2 channels are always inserted a little bit. And so water flows in normally and gets into the blood, but we're stopping that because we have hypertension. So we're stopping the addition of water. And because the glomerulus kind of sucks out water anyway, we start to get a decrease in pressure. So that's one way that it works. I know I'm way off on my slides, but we'll go through it again. The other thing it can do, it also inhibits these epithelium sodium channels. We just call them ENaC channels. Remember, those come from a different animal. Those come from aldosterone. And aldosterone is a different type of hormone than natriuretic peptide. That's a peptide hormone. Aldosterone is a steroid hormone, so that doesn't have a receptor on the cell membrane. That goes through the cell membrane, and its MR receptor lives inside the cytosol. And so it binds, and it still does its thing through the central dogma. It makes these ENaC channels. But now if we have ANP around, it will bind to its NPRA receptor. And through a secondary messenger system, it's an AMP system, I think. We don't need to know that for our level. But through a different set of messengers, it goes and it knocks out the implantation process of the ENaC channels. And so if you don't have ENaC channels, you can't pull sodium out of the filtrate and put it in the blood like we're usually doing. There's always a basal amount, a tiny bit amount of aldosterone around. So we always have a little salt and water coming in. And those are both cut off because of this ANP. That's the whole story right there, told on one slide. But now let's kind of back up and go through it again. So, and you could say that because, I know this is kind of good that I did this, because to understand this, it promotes natriuresis and diuresis. So natriuresis, nat means sodium, that means urine. So that's sodium in the urine. And it promotes that because it blocks the sodium epithelial channels. So sodium's trying to get out of the pre-urine or the filtrate or whatever you want to call it, the river water. It gets out normally, but not today, not if there's ANP around. So therefore, you're going to have kind of salty urine. And because it blocks aquaporin's 2 channels here, you're going to have diuresis. And that di means water in the urine, too much water in the urine. So you're also going to have the volume of urine is going to be too much, and it's probably going to be too salty as well. So in layman's terms, it kicks salt and water out of the blood, or it prevents it from coming back in. right? And this action, if we don't pull salt and water into the blood, because of the glomerular capillaries are sucking salt and water out naturally, the volume of blood fluid is going to decrease, and therefore the pressure is going to decrease as well. Okay, so here's everything I said. Whoa, way too far ahead. I should use my mouse maybe. Let's see, where were we? That's so far ahead. We're right there-ish, or right here. So yep, so everything we said, it appears to act as a, make sure you know this word, because that'll be on the test. And don't come up and say, what is that word again? I'll go, I don't know. An agonist. So an agonist, or an antagonist, not an agonist, an antagonist. An is the key word. An means it's going to bind to something like these V2 receptors and block their function. An agonist, if I erase the ant right here, just said agonist, that would bind to the V2 receptor and stimulate it to do something. So, and you know I'm going to test you on that, so I'll tell you right now, know the difference between an agonist and an antagonist. I should have put a picture of a little ant there. Okay, so that's what it does. It blocks the V2 receptor, interferes with the binding of vasopressin, therefore aqua 2 channels can't be put in, and therefore free water is inhibited from being absorbed or reabsorbed into the principal cell, which gets right into the blood. Okay, everything we said. So we said this, ANP also binds to natriuretic peptide receptor A. That's the NPR radio, NPRA. And it stimulates the secondary messenger system, which ultimately inhibits the insertion of ENaC channels into the apical membrane. Therefore, the sodium then stays in the filtrate and doesn't get sucked back into the blood. All right, backing up, I think we covered this already, but diuresis, that means water, urine. So you got too much water in the urine, a watery type urine. So the renal tubules and beyond have too much filtrate in them. It's usually water heavy. The excessive filtrate water causes polyuria. That's poly means too much. Urea means going to the number 2, peen. So too much peen, polyuria. And it depends on the cause of the diuresis. We'll look at different types. But the filtrate, even though you have diuresis, you're peeing too much. It could be a hypotonic diuresis. It could be a normotonic. What do you think that means, normo? The relationship between salt and water in the urine is perfect. It's in perfect harmony, perfect homeostasis. Or hypertonic. Hyper, I said to always look at the first, and hyper and hypotonic, and that means salt. So hypotonic means salt poor urine, too much water. Hyper means salt rich urine. The ratio between salt and water is very high. It's got too much salt in it. Okay, so what are the causes of hypotonic polyuria? So this is a patient who is urinating too much, and you test the urine and it's got too much water in it. It's hypotonic. So this is also known as water diuresis. Water diuresis. We know diuresis is really just like polyuria. You're going to the bathroom too much. And this just means it's got water in it. But same as AKA is hypotonic polyuria. Remember, hypotonic. Hypo means there's not enough salt and there's too much water. So water rules this one. So what causes hypotonic polyuria? Diabetes insipidus. And I can't remember if we talked about that much. I know I did in endocrinology. But we do know that vasopressin, we definitely know that vasopressin is released from the posterior pituitary magnocellular neurons. Pretty sure we've talked about that. And we know it binds to its V2 receptor, and it stimulates the implantation of aquaporin's 2 channels. And water just pours into the cell. And aquaporin's 3 and 4 channels are always in the basolateral surface, so water just pours right on out. In diabetes insipidus, this is broken. Very similar to ANP, breaking it. But there's the different types of it. There's a central diabetes insipidus, where the problem is the vasopressin is not made correctly, or maybe not made at all, or maybe it's made okay, but it can't get down. It can't be transported to the posterior pituitary like it's supposed to. So that's central diabetes insipidus. And then there is a renal diabetes insipidus, where the problem is right here. Either there's a mutation with the V2 receptor where it can't bind, or there's a mutation somewhere in the central dogma right here, somewhere in the gene or transcription factors, or maybe it just makes a mutated aquaporins 2 channel. So that's renal or nephrogenic. I think that's another word for it. I'm not going to test you on that, but I will when you get into endo. All right, primary polydipsia is another way, so let's explain that though. So if you take this out of action, the same story, you can't reabsorb free water, and so the salt to water ratio is very high water here. So high water or low salt, or high water, I said that right, high water and low salt would be a hypotonic solution. It's because so they have hypotonic polyuria. So that's one thing, diabetes insipidus. Another one is just primary polydipsia. So these are people who have mutations in their hypothalamus, specifically in their thirst center, which is the center in our brain that tells us when it's time to drink water. We're thirsty. Theirs isn't working, so they're constantly drinking water, way too much. And if you drink way too much water, you're always going to have too much water going into the urine, and the urine's going to be always sodium poor, water rich, so hypotonic polyuria. What causes hypertonic polyuria? So uncontrolled diabetes is the classic here. I always ask this on the exam. I think we've actually talked about this before, but again, maybe it was endo we talked about it. But here is let's say that's the connecting tubule of the renal tubules, kind of the distal part of the distal convoluted tubule. And there's a cell, call it a principal cell. And here's a peritubular capillary, and there's the interstitium. So in people with diabetes mellitus, when it's not controlled, they have glucose molecules in the filtrate. Normal person would have no green glucose molecules in their filtrate. People with diabetes, they can't get sugar out of the blood, and therefore the kidney ends up filtering too much sugar, shooting too much sugar out the glomerulus, and they can't reabsorb it in the proximal convoluted tubules. So you get this super osmotic, water-loving molecule. And you can think about this as albumin, or albumin story. Think of it as oncotic pressure, and that's exactly what it is. So we have a sucking force because of these green dots. There's too many green dots here and not enough glucose molecules. These are disappearing all the time. And so it would be great. We know that particles love to come together in equal concentration, so the green dots would love to go this way, but there are no glucose transporters on the apical surface of a principal cell, so there's no way for them to get out of here. But there are aquaporins channels, and there are other ways to get things across. But to get water across, because the next thing, if we can't equalize the solutes, then we need to equalize the ratio between water and solute. And there's way too much water compared to glucose on this side. And so we need to get more water in here to fix that problem. So it literally will suck water out of the cell, principal cells and alpha intercalated cells, beta intercalated cells, all the renal tubular cells. Water will be lost and sucked in to the pre-urine or the filtrate, and that puts too much water in there. And so you get a hypertonic polyuria. Why is it hypertonic? Salt's not being soaked in there, but remember, hypertonic, I use salt all the time because it's the number one osmolite that that you make these calls on. But in this case, it's the diabetes, it's the glucose molecule that makes there too many particles of solute in this filtrate. So that's why it's still hypertonic. In this case, glucose is the star of the show. There's too much glucose. So the sodium trick doesn't work there. But there's too many solutes, too many particles in this solution, and it needs to be diluted down by sucking water in. So, and that is oncotic pressure. Those glucose molecules cause osmosis. They cause the movement of water through aquaporin's 2 channels. And there's always a couple of them sticking around. You never don't have any of them there. Remember this. So just to bring back memories, now we can test on this again. This is our cartoon of the capillary. Remember your forces. The heart pushes blood through this capillary, and that pushing force is hydrostatic pressure. Hydrostatic pressure peters out as you go across the capillary. So it's high here, and it gets lower and lower and lower and lower down to like 15 millimeters of mercury here. So therefore, the pushing out of the capillary force is greater in the proximal capillary. Why is this proximal? Because the flow of blood is going this way. So if we pick a reference point here, or wherever you want to pick it, anything going that away is downstream, is distal. Anything going upstream is proximal. So this is proximal, this is distal. You could say this is arterial, this is the venous side of the capillary. But bottom line is the hydrostatic heart-generated force kind of peters out as you go across. But right here it rules. And so you push blood fluid out, which will replace this old interstitial fluid, glucose, nutrients, etc. But by the time you get down here, we have this sucking force. And this is generated by albumin. If you had tons and tons of glucose, it would help generate a force as well. But albumin, the glucose is sucked up by the kidney. So we have this sucking force, and that's always constant. That's because these little green albumin blobs, normally they don't get out into the interstitium. So there's always a 25 millimeter of mercury kind of sucking force, like the yellow arrows here. So by the time we get to the distal capillary here, you can see that the sucking force on the interstitium is now greater than the pushing out force. So therefore we get the sucking in of waste products and nutrients, and whatever else is floating around in here gets sucked in here. About 10% of the new fluid can't be sucked in here in the distal capillary. 10% is sucked in here into the lymph capillary. We all good with that? So with that in mind, so these pericapular tubules, let's remember this thing. So there's the glomerulus, there's the proximal convoluted tubule, loop of Henle, distal convoluted tubule. There's the thick ascending limb, which they didn't draw very thick, but they should have. And there's that late connecting tubule we talk about all the time. There's the collecting duct. But we do have a nest of capillaries that are just wrapped around all these tubules. And those are called peritubular capillaries. And those have the exact same forces going on in each capillary. They have higher pressure and they have lower pressure in the downstream part. And so that works at every cell level, that same story. This works at the pericapular tubule level here. So everything I said, I think. So, so that was the whole point of this. The whole point is to talk about pressure diuresis, which is probably a new word to you. So what the heck is a pressure diuresis? Well, diuresis is water in the urine, too much, but pressure means the pressure is the one that made extra water happen in the renal tubules. If you want to call all these tubules one word, just call them renal tubules. So there's too much water in here. So let's talk about how that pressure can work. And where does the pressure come from? Well, it comes from really high hypertension. So there's an artery let's call that the renal artery, that goes back to the descending thoracic aorta. Abdominal aorta is where it connects. If you have general hypertension, you're going to have really high blood pressure going all the way through here. If you have really, really high blood pressure, it's going to drive the water out like crazy in here. It's going to in hypertension, this force won't peter out. It's going to be super high all the way through because of the hypertension. See how that works? So the hydrostatic pressure will always rule, and you'll have a hard time sucking in extra water. No problem pushing it out, but it's a hard time sucking it in. So that's kind of where we're going here. So increased hydrostatic pressure in these peritubular capillaries can discourage the uptake of water. So even if antidiuretic hormone, it's not drawn in here, but so let's see, right in these connecting tubules, you'd be trying to suck water out of the filtrate, and you have another force pulling that back. Even if ADH is there and it's implanted aquaporins channels, you can't pull it. So let's look at this. So if you had super high pressure in this capillary, there's no oncotic pressure over here to pull fluid back in. Okay, so even if there's an aquaporins 2 channel and the water tries to go down it, it's not going to work because there's too much hydrostatic pressure and it's pushing it this way. There's no oncotic kind of sucking force here. It's all hydrostatic pressure. So therefore, it basically stops the influx of water because of a pressure gradient. See how that works? So yeah, so therefore you get a water diuresis and even a sodium diuresis. The sodium doesn't even like to, we go back to this. The sodium has trouble going against a pressure gradient because the sodium normally would get sucked in the distal capillary, but now there's no sucking force. So the sodium is blocked and the water is blocked in a pressure diuresis. See how that works? Yep, therefore you get a water diuresis and a sodium diuresis. And the water can't be reabsorbed out of the filtrate. That should be can't, cannot. Okay, this will give you what's called a pressure diuresis-induced polyuria. So pressure-induced or pressure diuresis-induced. Diuresis is too much water in the tubules. Could be any combination of those words. So it's just putting in physiologic speak what I just said. There's too much pressure, and therefore you have diuresis. The water can't get out, and if the water can't get out of the tubules, the renal tubules, then you have too much water to pee out in the toilet, and you got polyuria. See how that works? Probably be. If it's doing this live, there's probably be questions, hands going up, or I can see that look in some of your eyes, but we can go over this again when I get back next week. So yeah, that's everything we said already. Hypertension causes increased renal afferent arterial. Oh no, this is a different thing. This is another way that it works. So that's just one mechanism, but here's another mechanism. Okay, and I don't think I told you about this, but angiotensin II, there's always a little bit of angiotensin II floating around, and it loves to bind to the cells of the afferent arterial, and it loves to kind of vasoconstrict this a little bit, which would raise pressure in here. So if you had hypertension, that would turn off the R2A system. And so if we turned off the R2A system, there'd be no angiotensin II to bind here and constrict this. It would pop wide open. So you would have a ton of blood flying down here. More blood than normal would be flying down here to these peritubular capillaries. And therefore, if it's too much blood, there's too much hydrostatic pressure. And then everything that I just said works. So that's how the hypertension passes. We didn't go that in renal physiology. I'm sure Dr. James will teach you more about this. But normally the pressure is pretty even here all the time because we can regulate it right here with angiotensin II. But if angiotensin II is turned off because you have hypertension, then you get too much blood flow in and around these peritubular capillaries, and therefore it's really hard to suck nutrients in because the oncotic pressures are neutralized because the hydrostatic pressures are so high. Does that make sense? Right, so yeah, you can't reabsorb water into the blood. It stays in the filtrate, and then you have a diuresis. You could say it's a pressure-induced diuresis or pressure diuresis-induced polyuria, whichever way you want to say. Okay, so hypertension in these peritubules, pericapillary tubules, or peritubular capillaries, is the reason for this pressure diuresis. So one of the reasons, and we talked about the other reason too. What about natriuresis? So we kind of talked about that already. The nephron normally excretes, or if the nephron excretes too much salt into the filtrate, then your urine will have too much salt in it as well. If you have too much salt in the urine, you have a hypertonic urine. I think we know that already. So that's one way to get a natriuresis. The nephron is over-releasing salt, or not pulling or reabsorbing enough sodium chloride out of the proximal convoluted tubules. Letting too much salt out, it shouldn't do that. It should let some stay around. Another way that you can do that is if the nephron can't reabsorb the salt, because we know in the principal cell, the principal cells are always reabsorbing through ENaC channels, are always reabsorbing some salt. See how that works? Okay, and if you have a salty filtrate here, or river water, or pre-urine, whatever you want to call it, those extra sodium molecules act as osmolytes. And so you have kind of a sucking force. There's not a lot of channels that will take sodium out of the filtrate this far down in these distal convoluted tubules. So it acts like, you might as well consider those kind of like a sucking force, an albumin force. And it literally tries to suck water, not out of the filtrate like it's supposed to, but it holds water in. And then you get too much water on top of the natriuresis. See how that works? All right. We already know this, don't we? What stimulates the release of natriuretic peptide and brain natriuretic peptide? All of this is, or some of this is true for BNP. Again, we don't know tons about that one. So any of these factors will cause hypertension. Basically, anything that causes hypertension or anything that stretches the atria will set this off. So we have hypervolemia, we have hypertension, atrial septal defect. Have we talked about that? That's a hole in the intraatrial septum. I think we have. And when systole, atrial systole occurs, it normally shoots blood down here. But if there's a hole here, it injects extra blood in the right atrium. Guess what that does? It stretches the walls of the right atrium. So what? So you will release ANP because of that stretch. Little kids with ASDs will have elevated levels of ANP. Heart disease-induced natriuretic peptide. What's the story with this? So the overall theme here is if the heart acts as a beaver dam and it causes a backup of blood into the atria, it's going to stretch the atria. And that is going to shoot out atrial natriuretic peptide. So myocardial infarction affecting either ventricle, right or left, because both atria have cells that produce natriuretic peptide. So if you injure the ventricles, they can't pump blood out. So myocardial infection of ventricles, they can't pump all the blood out of the heart. So you have too much blood left over. The afterload is too high. Too much blood left over. And therefore, when the next cycle occurs, tons of blood squeezes into the ventricles and over stretches the ventricles to the point it backs blood up into the atria, and the atria get overstretched. And that overstretching of the atria causes the release of ANP. Heart valve damage is the same thing. If you have a mitral valve stenosis or even aortic valve stenosis, and that can apply to the semilunar valves and the pulmonary valve as well. If those valves are not working right, again, you're not going to be able to empty the left heart completely. And so when the next cycle starts, you're going to overfill the left ventricle, and some of that blood is going to overfill the right and left atria and overstretch them. The beaver dam is the ventricles, and the traffic jam and the backup affects the atria, then it goes into the lungs, and we know that whole story. So overstretched atrial cells release ANP. Same theme on all of this, ischemic heart disease. So that's atherosclerosis of all those coronary arteries I made you memorize. Remember those things? That will cause the myocardium to start to die and fibrose and scar up, and basically ruins the pumping function of your ventricles. And if the ventricles, just like with a myocardial infarction, if the ventricles aren't working, you're going to have extra blood left in there, extra blood left over from the last cycle, and when you try to refill the ventricles with a new cycle, the blood backs right up and goes into the atria and overstretches them, and that releases ANP. See, anything that causes heart failure will do that. Cardiomyopathy is a fancy word for heart failure. Even more, what about the kidney? So the kidney can definitely cause the release of ANP and BNP. So how does that work? So let's look at chronic renal failure. So and someone with kidneys who are failing, they can't get rid of the salt or the water. They're so broken that the glomerulus can't push out the salt and water into Bowman's capsule for it to become filtrate or pre-urine. They don't work that good. And so you always got too much sodium and too much water in your blood, and that right there is the definition of hypertension, and that's going to stimulate. Just because when you have hypertension, there's too much blood stuffing into the left ventricles. The incoming pressure is too high, and it overstuffs the right and left ventricles and atria. Stretch of the atria will do it. The nephrotic syndrome, nephritic syndrome, you also can't clear sodium and water from the blood. Same story. COPD and pulmonary hypertension. So the respiratory unit we talked about somewhere. The alveoli. So there's diseases, COPD, chronic obstructive pulmonary disease, which is usually a combination of chronic bronchitis and emphysema. They inflame the alveoli in the microcirculation around the alveoli, so it makes it difficult to push blood through the lungs. So blood backs up into the right heart, the right ventricles at first, and then it backs up into the right atria and stretches it, and there you go. The stretch of the right atria will cause the release of ANP. Chronic tachycardia is like atrial fibrillation, where the atria are beating 550 beats per minute. That constant beating will irritate those cardiomyocytes so much that they just start increasing the production of ANP. Plus the pressures in a heart beating that fast are very high. So study tip, if you have too much pressure in your arteries, that's hypertension. Then you're going to have too much blood that is going to get stuff into the aorta with every beat. I don't know what I'm trying to say here. What am I trying to say? Too much blood is going to get stuffed. Oh, if you have hypertension, the pressure coming into the heart is too great, and it's going to stuff too much blood into the heart, into the atria and the ventricles on every beat, and that will stretch the atria and stimulate the release of ANP. Okay, here's just another kind of other actions of ANP. We won't go into the crazy details of these, but you can see it has just about the opposite actions of angiotensin II. It can bind to magnocellular neurons and cause a decreased release of ADH. It can bind to glomerulosa cells in the zona glomerulosa of the adrenal cortex and inhibit the release of aldosterone. It also binds to both of these, or it binds to ADH's receptors. We just talked about that. It can bind to the afferent arterial cells, specifically the juxtaglomerular cells, and flat out decrease the production of renin. Because we don't want renin. Renin will try to increase blood pressure. So it can do that as well. It also can interfere with sympathetic ganglia transmission. So bottom line, it decreases sympathetic input to the kidneys and heart, which would stimulate the heart to, well, if you decrease that sympathetics, it would slow the heart rate down. Remember, sympathetics speed up the heart. And kidneys, it would slow down, or it actually would speed up glomerular filtration rate, which would lower the hypertension. It increases parasympathetics to the heart, which would also slow down the heart. Causes vasodilation of the ascending aorta, which is going to decrease the blood pressure. If that lumen is wider, then it could absorb that blast of blood out of the left ventricle. And it also causes general arterial vasodilation, which will also decrease blood pressure. Some fun facts: half-life is only 3 minutes. There's some clearance we don't have to worry about this stuff. Its release is increased by exercise. I think I do test on that one. Exercise is so darn good for you. There's so many receptors that are created and amplified, and so much good stuff comes from exercise. So if you exercise, it stimulates the release of ANF, even if you don't have hypertension. So it lowers your blood pressure, not dangerously low, but especially if you have hypertension, the first before you start taking meds is get some exercise. See if you can't naturally lower that just by changing your living style. Yes, it's always secreted at the low, kind of a low basal level. The gene for ANP is found on chromosome 8. BNP, let's talk just a little about that. This is very confusing. People like 30% got this one right, even though I warn you it's going to be on the test. Brain natriuretic peptide, known as BNP. It could be called brain natriuretic factor as well. Don't know as much about this one, but it's not produced in the atria. It's produced mainly in the ventricular myocardial cells. It's also stimulated, just like ANP, it's stimulated by increased pressure and stretch of the ventricle. So all those conditions that increase blood flow overstuff the ventricles with blood. This will be released in addition to ANP. So all those conditions still apply to this. Here's the one that everybody misses. So brain natriuretic peptide, it's made in the ventricles. It's not made in the brain. So they originally discovered this in the pig brain, and pigs have very similar organ function. Their disc function is very similar to human. They have a lot of similarity to humans, and they just assumed because they discovered it there, that's where it was produced. And years later they found out, nope, it's not produced there. It's actually coming out of the ventricle of the pig. And then with human research they confirmed that. So physiologically it's almost identical to ANP, so I won't go through all that. The gene that releases it, it's the same type of gene, it's just coming from a different chromosome. It's less responsive than ANP to abrupt changes, so it's more powerful than ANP, so if it is triggered, it's a more powerful lowering effect of blood pressure. Like exercise doesn't seem to set this one off. Exercise seems to release ANP, but not BNP. So it's less responsive. Its trigger is, you got to hit it more to get it to fire off. Myocardial infarction, again, causes huge amounts of BNP to be released. And we've talked about overstuffing already. and AMP to be released. Half-life is longer. It's about 20 minutes. It doesn't bind. It binds less aggressively to NPRC receptors. I cut all the receptor stuff out because we're getting too deep into the weeds. So I'm not going to ask you about receptors, about NPRC receptors anyway. Should know the NPRA receptor, which is on the principal cell, created by a gene on chromosome 8, lives right next door to the AMP gene. And we know a little bit about type C or C-type natriuretic peptide. Sometimes it's called CNP or CNF, C for central nervous system, and that's where this comes from. So it's expressed by cells that live in the central nervous system. Some endothelial cells, blood vessels, can also make it, but mainly in the central nervous system. We won't worry about where. Just it's pretty ubiquitous. Pretty much all of the neurons can secrete it. It's not secreted in the heart, though. The function still isn't well understood. It does cause vasodilation of arterioles, so that's its blood lowering or hypertension fighting effect is to vasodilate arterioles. It has something to do in pre-adolescence of bone growth and bone elongation, so it affects the long bones. It's not considered a true hormone, though, because it acts more on the local tissues when it's secreted. So it acts as an autocrine and paracrine. So it's kind of a weird. You think of it as kind of complementing the actions of AMP and BNP. Okay, similar amino acid structure. Half-life's about 3 minutes. I'm never going to ask you the half-life, exactly what that is. Currently not a whole lot known about this. And it doesn't have really a natriuretic effect on the kidney. Its natriuretic effect is on the arterioles. So it doesn't bind to principal cells like AMP and BNP do. Okay, there's just a gene. I'm not going to ask you that, but it's the same polypeptide change. You can see where it's cleaved by proteases to make ANP. They're calling it A and F in this diagram, BNP and CNP. I would never ask you that. That's only 38 slides, but I bet your brains are spinning. You're probably full. One of my favorite pictures. I wish it was sharper. Questions, email me the questions. Like I said, if all goes well, I should be back on Monday, and we'll see you later.